Good morning, everybody. This is the third Data Bliss presentation, which is as part of the summer program. And we have 3 speakers lined up for today. The first speaker is Dr. Chung-Wai Chow, who is an associate professor and clinician scientist in the Division of Respirology with a cross-appointment to the Faculty of Engineering and Applied Sciences and the Dalalana School of Public Health. Her research focus is on the biological effects of air pollution on airway inflammation and chronic inflammatory lung disease. Dr. Chow.
So thank you very much, and thank you for the invitation to speak. When I was thinking about how to do this data blitz, I decided that it may be more entertaining and perhaps more informative for you to actually give you an overview of my research program through the lens of IMS students and the students who've actually contributed to the research, as opposed to a deep dive into the actual science of the work that I do. If you want to do that, you're welcome to join my lab and come to our weekly lab meetings. And really, as mentioned before, I am a clinician scientist, and my research is really a very iterative process. I went into medical school knowing that I wasn't going to go out and set up a private practice. I was looking for something else that I could contribute to the field. And over the course of the years as an undergraduate student, as a medical student, and actually as a resident, I did all sorts of different types of research to figure out what I really wanted or didn't want to do. And actually the easy decision was really figuring out what I really disliked before I figured out what I really wanted to do. Now, my association with the IMS goes a long way. And in fact, after I finished all my clinical training, I decided to go into to go back to school and to do a graduate degree in cell biology. And I was the very first trainee in the clinician scientist program. And Elliot Phillipson, whom you see here on the left, is actually a superb scientist, a great man, and an Order of Canada. And I'm very proud, actually, that as the first trainee in this program, when he had a celebration to celebrate his Order of Canada appointment, I was actually invited to that celebration. And 2 other really key people in terms of mentorship for me early on is Greg Downey, who was a former director of the IMS, and Sergio Grinstein, whom I think some of you will know is actually a superb scientist at the Hospital for Sick Children. And so that was my basic research training. I actually, after I finished here, I did a 2-year postdoc at the Max Planck Institute to learn molecular biology. And subsequent to that, I returned to Toronto, and in a transition period that some of us who are faculty members know, it was somewhat chaotic. At the time when I arrived, there was no space. There was a building boom in Toronto in terms of building of institutions, but what that meant was that there was actually no physical room for any new faculty members. So I actually started out my lab in a little bench space in my former mentor's lab, and my office space was literally the entranceway to Elliot Phillipson's lab. And so I actually got to know him really, really well. But during this period of time, and I was actually, I was sort of in this quasi-state where I was faculty member, but not really, where I was trying to sort things out. And I was actually assigned 2 summer students who were IMS students. And some of you are probably primarily supervised by postdocs or graduate students. And don't take that actually as an insult or any diminishing of what your role is, because actually as a postdoc, as a student, sometimes they have much more time to devote to you, to devote to your learning than the actual PI may have. And I can tell you that surely is the case as my career has evolved. And 2 notable students during this period of time, Christina Sheck and Kalpana Sharma, actually was back in the lab several times. By the time that I was assigned as their primary supervisor, that was their 3rd summer in the lab. Christina is now a drug inspection officer with Health Canada, so she's the person who's actually responsible for making sure that things like shipments of Tylenol, baby powder come in are really what they say they are, and they're not fraudulent. Kalpana Sharma subsequently went on to medical school, and she is now an obstetrician at North York General. So these are the 2 ladies here. And And you will hear a little bit about Andre Chow. He will come up in a little bit later. And it was during this period of time, sort of in terms of thinking about what my research program was going to be like. All of us have somewhat grandiose ideas about what we want to do when we start, and the reality hits is that there's you and a little bench space and a little desk, and if you're lucky, you have some students working with you, and then the process of hiring people, which none of us are actually trained to do, occurs. And during this period of time, I really drilled down back to actually what I learned as a PhD student, and went back to really very, very basic cell biology, molecular biology techniques. And it doesn't really matter at this point what it is, but at the time, it was thought that, and remember this is about 10, 15 years ago, it was thought that inflammation was all being mediated by professional inflammatory cells, neutrophils, macrophages, lymphocytes, and that the other cell types, the airway epithelium, the endothelium, really did not have very much role. And during the time that I was a postdoc, I was working on this tyrosine kinase called Syk. There was all sorts of disparate information that was telling us that Syk was actually important in other cell types, and it may be important in airway epithelium, in inflammation, but nobody was really paying a whole lot of attention to it, which is terrific, because that meant that was something that I could focus on. And this is really the very first research focus in my laboratory. We used at that time very state-of-the-art, at that time was state-of-the-art techniques, which was immunofluorescence, looking at the localization and expression of Syk in airway epithelial cells. We were not only able to show that it was there, but that actually when we stimulated the cell with human rhinovirus, which is the cause of the common cold, we were able to show that it actually localized to the airway, to the membrane of the airway epithelium. We subsequently went on to use molecular biology techniques to show that there was actually co-localization, and that actually using biochemistry techniques that they actually co-associated with each other. And lastly, what we did was we actually looked at functional outcomes, and we downregulated Syk expression by using this siRNA. And when we knocked out Syk expression, what we found was that it actually decreased the amount of interleukin-8 that was being expressed. Now, this work was mostly done by a technician who's still working with me, Chow, but it certainly would not have been possible without the help of some of the summer students who worked with us. Andre Chow, whose picture you saw before, and Christina Lau, who was a summer student before becoming my very first master's student. And so over the course of several papers, we were able to establish the function of Syk in airway epithelial cells, how it was regulated, where it was, and what it could possibly be doing. And Christina remains to this day probably the most successful of my graduate students, and that's only because the others have not had time to become successful yet in the ways that Christine has. She was quite prolific. She was the first author in 2 papers, co-authored several papers, won numerous awards during her time with me. And she was just appointed on faculty at U of T this July. And what was remarkable was that she told me that she was appointed primarily because of the work she did as a graduate student. There was quite a large field of people who were being interviewed, and she kept insisting that her master's work had absolutely nothing to do with clinical care and palliative care medicine, which is what she's interested in. And what they said to her was that your master's, you learned how to think during your master's, and the fact that you published, and we know from the awards that you've won that actually you know how to think on your feet and you know how to be agile and think. Now, and so Christine was my very first graduate student, and this was at this time, basically the end of one of my first grants. And at that time, I was getting a whole lot of feedback that it's time as a clinician scientist, I've moved beyond cell types and to look at something a little bit more holistic and look at some animal models. And at the same time this was happening, there was a lot of buzz in the field about Syk inhibitors as potential therapeutic agents. It was being tried in rheumatoid arthritis. And at this time, I started talking to some of my lab neighbors. And it was through this sort of collaborations with people in the scientists at Dalla Lana and subsequently in chemical engineering, that we began to look in the field of airway inflammation and air pollution, and couple that with my interest in chronic airway diseases. But what it also meant was that there was a lot of traveling back and forth. Not so much for me, but actually for the students who were working in the lab. At that time, when we first started out, I was still here at the MSB, but we were doing a lot of our research at the Gage Institute, which is down the street. And then subsequent to that, I actually moved to TGH Mars, and there was even further traveling involved. And the movement into sort of in vitro cell culture models into animal models really began with the second graduate student who was also an IMS student. She started out working for me actually as a research assistant and transitioned into a graduate student. And what she did was she established in the lab an animal model of airway inflammation or of asthma. At that time, we were using a 12-week model. And at the end of this 12-week model, where we actually had an asthmatic phenotype, we took the mice and actually took them to the Gage Institute and actually exposed them to college street air, concentrated air pollution at street level during the summertime, mostly. And at the end of that, she treated the mice with a Syk inhibitor that we were able to obtain from one of the drug companies. And what she was able to show, so at the end of the treatment, she gave it drug inhibitors. What she was able to show is that in the asthmatic mice, which showed an increase in airway hyperresponsiveness in comparison to the control mice, which were the OVA PBS, that after exposure to college street air, there was a significant increase in airway hyperresponsiveness, which for an asthmatic means increase in symptoms. But when she treated the mice with the Syk inhibitor, she was able to downregulate the expression. Or the airway hyperresponsiveness, showing that actually Syk actually played a very important role in terms of airway hyperresponsiveness and possibly in airway inflammation. What she also showed in this paper was that Syk was actually highly expressed in the airway epithelium and much more so in the asthmatic model. And in sort of later studies that were done by two other master's students who were also IMS students here, we went on to look at the role of Syk as a chronic treatment for asthma. We were able to show that, in fact, that we were able to decrease airway hyperresponsiveness, but we weren't able to actually decrease in airway remodeling. Unfortunately, or fortunately, depending on how you look at it, about the same period of time, a lot of the pharmaceutical companies' interest in Syk inhibitors actually waned. Clinical trials at that point had showed that there were some side effects that were, unexpected, and the Syk inhibitors had been pulled off. And while we were doing this, Serena Zhang is the last of my graduate students who have since graduated, and she was also looking at a model where we were actually using a chronic model of asthma and chronic exposure to look to air pollution to look at the effects of ongoing air pollution exposure in asthma. Both Sapir is still working with me as a research coordinator, and he's transitioned from working with animals to now actually working with me in a number of clinical studies. And Serena, some of you probably will know, she may have run into her because she works for now one of the companies that supplies scientific supplies. And as my sort of research had sort of evolved from initially, actually as a clinician, looking at sort of basic questions that I wanted to answer in sort of at a very basic level at the bench, went from cell culture models to animal models, and more recently we've moved into actually human models and human trials. But a very big part, as I mentioned before, a big part of my thinking process is always very iterative. I always want to figure out, what is the basic mechanisms that's responsible. Can I answer this in a clean way in an animal model? So a big part of my lab still works on animal models. Dave Gull and Lindsay Wu are two master's students who are continuing a lot of the work using different models of inflammation, chronic diseases in the lung, asthma and cystic fibrosis, and the effect of air pollution. And I'm not going to actually mention very much in terms of the three IMS summer students who are working in the lab this summer. I encourage you next week to go to their posters and talks and hear what they have to tell you about their summer with me. And lastly, I mentioned that research sort of has taken me through an unexpected journey in terms of some of the things I wanted to do. I spent most of this July in Fort McMurray, and this is actually a picture taken right across the street from the Suncor oil sands operations. We have several ongoing studies that's looking at actually the effect of both the wildfires from last year's fires on the inhabitants of Fort McMurray, and also in looking at the effects of oil sands in the region and working with the First Nations up there. I will be recruiting students next summer and graduate students, but as some of the summer students who work with me know, I have absolutely no idea when you come to talk to me in the fall what happens the following summer. And that is probably the most wonderful thing about research and the most wonderful thing about what we do.